Chapter 8, Microbial Genetics. So we are going to start with some terminology, which I think will be mostly review for you. We will then go into the structure and function of genetic material, DNA, and look at some of the differences between prokaryotes and eukaryotes as they replicate their DNA, as they use their DNA to make mRNA and then translate that mRNA into protein, and then also ways that they regulate expression of the genes within their DNA. And you'll see that there's not too many differences that we're going to focus on. And so I do think that with what you've already learned in previous courses, most of this will be review for you. And then we're going to touch on mutations. What are they? How do they happen? What does the cell do about them? And then we will very briefly look at ways that genetic changes can occur not from mutations but through what we call recombination. And so different sources of recombination can include conjugation, transformation, and transduction. So this, I just caught this weird typo here. It says recombination. Really, recombination should be above, and then down here should say conjugation, transformation, and transduction. Now, as far as the specific steps of transduction and conjugation, we will see that in lab. So I'm not going to test you on the mechanisms of horizontal gene transfer. For Chapter 8, I just want you to know what is recombination, what are the 3 types of horizontal gene transfer that we're focused on in this class, right? And we'll even look at one experiment that led to understanding that transformation, what transformation is, and how, that it can happen. And so that's it. So this section where we have this line drawn is going to be super brief. You're also going to have a separate video for horizontal gene transfer. That's why I put C lab recording down at the bottom. And the lab recording is in next week's module. And that will go over the mechanisms of horizontal gene transfer. So that will be part of your lab. And then that will be tested in exam 3. Okay? All right, so let's go ahead and get refamiliarize ourselves with some terminology. So genetics is the study of the genes in an organism. So what genes are, how the genes are acting as a source of information, how do they carry information from parent to offspring, right, heredity. How is that information utilized? How is it expressed into a product? How genes are replicated? How does the cell replicate its information to produce offspring, right? And then how is the expression regulated within the cell, and furthermore, how is that regulation transferred from cell to cell so that you can maintain the same traits, characteristics, right? Okay, so that's genetics, big field, right? The genome is all of the genetic information in a cell, right? And a gene is just the small segment of DNA, so a particular sequence along the big molecule, taking just a small sequence that from beginning to end of that sequence is what leads to the formation of a product. And most of these products will be protein, and that could be an enzymatic, right, enzyme protein, or it could be a structural protein, okay? Now, in eukaryotic organisms, we'll see that there are also other proteins that are not exactly an enzyme and not exactly a structural component, but there can be other functions but we won't really see that in our focus today. Okay, chromosomes, structures containing DNA, and this will be the organism's genetic material. This is the unit that's copied and passed on to offspring. And for prokaryotes, their chromosome is relatively simple, relatively small, and it is circular. So it's one circular chromosome. It is a double helix still, and then that will be supercoiled so that it fits within the cell. And we also will see that they will anchor at one or more points. Their circular DNA will be bound to the plasma membrane to kind of keep it organized, keep it in a specific area. All right, eukaryotes, on the other hand, you know that we have a nucleus that contains our chromosomes. A eukaryote will have one or more linear chromosomes. So they just look like, when you look at them, the classic example, right, that you're used to seeing is the little X, right, which would be two copies of the chromosome before cell division. Okay. And of course, these chromosomes are highly organized structures that have lots of different proteins involved to keep it organized and highly condensed. Again, that classic little X that you're used to seeing for chromosomes, kind of this kind of, gosh, it's really hard to write with the mouse. Sorry, guys. Okay. There. You're used to seeing something kind of like this, right? That looks almost like an X. And this is one chromosome, and this is a copy of that chromosome. And then they will be separated when cell division occurs. And we'll just ignore this ugly mistake here. Okay, so there are lots of proteins that are holding it into this super condensed form. It doesn't stay in this condensed form throughout the whole life of the cell. In fact, when it's super condensed like this cell cannot even get to it, cannot utilize it to make anything. So no, what we call protein synthesis, right, won't be able to occur during this highly condensed form. So we actually only see this right before cell division, and then it will start to uncoil. It'll still be kept in a slightly, condensed form, but it won't be condensed enough for us to see under a microscope any longer. So that's something kind of neat to think about when you look at cells under the microscope, eukaryotic cells, and you see those classic chromosomes, you'll know it's about to undergo cell division, or it's in the process of undergoing cell division, because at any other point in its life cycle, you won't see those classic chromosomes. All right, so genotype refers to the specific sequence, the actual genes of the organism, right? The code, the sequence, the ATCG pattern, okay? The phenotype is what you observe, the product of that gene. So classic examples that we give are with people, and we say like hair color, height, right? Widow's peak or not, rolling your tongue or not, eye color, all these different, attached or detached earlobes, all these different traits that you can see physically is the easiest to give as an example, but that it's not limited to what you can see, right? And especially when you're talking about single-celled organisms, you would be talking, phenotype could be whether or not it can eat a particular type of nutrient, whether or not it produces a particular product, and so you're looking at one or more products from a gene, so at the molecular level, right? So expression of the genes, sometimes referred to as the traits of the organism, or thinking about the properties of the organisms, which are derived from the genotype, right? Okay, so gene expression, as I've already mentioned, is taking a gene, the coding sequence, transcribing it into mRNA, right, messenger RNA, and then translating it to a functional protein product. So you translate it from the nucleic acid, mRNA, into the language of protein, which would be amino acid. And so that's what we mean by the gene is expressed. So the flow of genetic information within the cell, you have using it to express, right, what we just talked about, transcription of a gene, and then translation of that transcript into a product. So that is happening almost always, with the exception of cell division, right? Then we have recombination events. This is that bottom part of the outline that I showed you, where I refer you to another recording for horizontal gene transfer. It is not limited to horizontal gene transfer. It also includes transposon, which we'll briefly mention at the end of this lecture. So this just means any method to get a new combination of one or more genes, right? So you have to have some donor DNA, some DNA that was not originally part of the cell, and then or at least not originally in that order in the cell, and then something brings about a change, and you get a new series, a new sequence of DNA, which leads to some new function, right? Usually it's a gain of function, okay? And our main concern is with antibiotic resistance. Okay, so this will be at the end of the lecture and then also in that other video. Okay, replication is what we're going to start with today, looking at taking all of the genetic material, making a copy, and then giving that copy to a new cell, a daughter cell, and the daughter cell will be identical to the parent cell. Okay, so this is what we're going to look at. So the flow of genetic information to a new cell is replication. The flow of that information being used to make products is gene expression, which will be we'll look at immediately after replication. And then recombination is forming a new sequence and a new function, which will be at the very end of the lecture and in that supplemental video. Okay, so DNA is the blueprint. All of these start with DNA, right? All of these different types of flows start with DNA. DNA is the blueprint. So bacterial chromosome is different than eukaryotic chromosome in that it's circular, right? They are both double-stranded DNA, so that's the same for both. And then for the bacterial chromosome, we say that it's supercoiled, and we don't really use that term for eukaryotic DNA. And as I mentioned earlier, the bacterial chromosome can be attached at one or more points along the plasma membrane. And then something new that you might not know is that the chromosome, it does not have continuous coding from beginning to end. It will have a gene, might have several genes in a row, but then there will be these regions that are non-coding regions. They don't code for anything, and they will get removed, or they just won't be copied into the mRNA. So for the way we think about it, they're useless for the cell. I'm sure that there's a function that we will learn about at some point in time. It's hard to think that anything would be completely wasteful and still remain, right? But as far as what we know so far, these little short sequences, they're 2 to 5 base pairs in length, right? So base pair being A, T, C, or G. They are 2 to 5 base pairs in length, and they're repeated over and over again for some length. And we call this a short tandem repeat because it will be, right, a repeated series, and it doesn't code for anything. It seems to be in between genes, so it separates coding regions. And as I mentioned, we don't know what its function is right now. It doesn't seem to have an obvious function, but we've come up with a way to utilize it for our benefit. So we can utilize it for an assay called a DNA fingerprinting assay. And for microbiology, the main focus for DNA fingerprinting would be to quickly be able to identify pathogens, right? But in the broader scope, it can be used in all sorts of ways, such as paternity tests, right? If a woman says, you're the father and want support, at least financial support, to raise this child, and he says, no, I'm not, I do not believe that I'm the father, they can both give DNA samples, and a DNA fingerprinting analysis can be done, and then say yes or no, right? So you would need to take the mom's DNA, the father's DNA, and the child's DNA. The child should have half of its DNA identical to mom and half of its DNA identical to dad. So in that way, we can determine who the father is. Okay. We can do crime scene investigations in the same way, or similar way. So you take samples from the crime scene, and then you take samples from your suspects, and you should have 100% match for the DNA at the crime scene to the DNA of your suspect. If your suspect is the person that was at the crime scene that you took, you have the sample at the crime scene, it should match 100%. Now, the shortcoming with that being that all that does is says that the person was there, right? It doesn't definitively tell you that they did anything in particular, right? And so then detectives and such would have to construct the rest of their case to, provide motive and all the other evidence that would then actually convict the person. We actually offer a forensic science course here. It's not transferable to an actual forensics certificate, but it's a great way to learn about all these modern tools, and then you would see if you are interested in going into forensics. So I encourage it. And if not, just to have a very interesting course that will really strengthen your molecular skills, and it can be very beneficial for any of the biology degrees that you pursue. Okay, because a lot of the tools that we use in forensic science are also used in and applied in different ways in all sorts of different fields. All right. So that's my little promotion for our forensic science class. We also have a human heredity class, right? And we've been talking about, our focus today with DNA replication and passing that on to your offspring is heredity. We won't be studying traits in this class. When you take if you take a biology course, you'll have a little section on Mendelian genetics, and you might even go a little bit further. If you do 4A, our majors biology course, then you will get to do a big section on genetics. And if you're really interested and you want to understand more about human heredity, then I would encourage you to take that course, which is usually taught by the same instructor who designed the course, Al, and for some reason I'm blanking on his last name. I think Gonzalez, right? Al Gonzalez. Very, very nice and fun professor. So those would be two really good elective courses to enhance your biology skills. I encourage you to think about. All right. So the prokaryotic chromosome I mentioned is supercoiled. If you were to, kind of poke the cell and allow the chromosome to unravel and come out, you would see that it's very, very large compared to the cell itself. And so that explains, right here in this visual, you can see why it would need to be supercoiled in order to fit inside of that cell and also have space inside the cell for other things like ribosomes and such, right? And then on the right hand side of this image, if you were to take this entire chromosome and unravel it to be a complete circle, then you would see something like this. And I do not need you to memorize this actual structure here. I just want to demonstrate, give you a little perspective on bacterial chromosomes. And so starting at the beginning, which would be the origin of replication, the OREC, we start base pair 1 and we move all the way around, and here we're keeping track of base pairs by the thousands, right? So we have kilo base pairs going around. And so these different colors along the chromosome show you the type of genes that are present in that area. And notice there's a lot of space in between some of these genes, which would be full of those short tandem repeats that we saw a second ago, right? And so all the blue are the genes that code for amino acid metabolism. All of the reddish orange color is carbohydrate metabolism. We see the membrane synthesis is interesting. Membrane synthesis, all of the genes are clustered together in one long stretch. And that's everything, all the genes that are needed to make the membrane, the plasma membrane, right? Then we have the lipid metabolism, so to be able to take in nutrients and get it ready for membrane synthesis is in green, and it's just here, right? And then we have DNA replication and DNA repair, which is scattered across. So here, these, and here. And so that's it. You don't have that much, right? It's relatively simple and relatively small. And you might be thinking, well, how would I know? Because what else do I have to compare this to? So I've given you a little comparison here. If you were to take the genome of the bacteriophage virus, you would see that it is made up of under 200,000 base pairs. So we're in the thousands, hundreds of thousands of base pairs. Then, if you move to bacteria such as E. coli, you are looking at 4.6 million base pairs. So you're in the millions of base pairs for the entire genome. Now, this is including the non-coding sequences in addition to just the entire genome. You're not just looking at the genes, but the entire base pairs of the entire genome. And then the fruit fly, right, just a fly, has 130 million base pairs, much more than the bacteria, but much less than humans. Humans have 3.2 billion base pairs. And interestingly, plants can actually have more base pairs than people. And so this, even though we think of this increase as you get an increase in complexity, that idea kind of breaks down when you get up to the top, right? Because obviously humans are more complex than plants. We are the highest organism on the tree of life, but But the plants, for some reason, have more genome base pairs. Okay, so this is fun. You do not need, there's no need for you to memorize these numbers. All right, so bacterial DNA synthesis is pretty similar to eukaryotic synthesis. Key difference is, of course, that you're working with a circular piece of DNA, and you have what we call an origin of replication, a single origin of replication. Okay, so that's a key difference. Now, in both of the DNA replication for bacteria and eukaryotes, you have to have topoisomerase to relax and start to open up the DNA. You would need a helicase to bind and separate the two strands, and you'll need to have an RNA polymerase to make it's special RNA polymerase called primase, and it makes a short sequence called a primer. And then you would have DNA polymerase 3 come in and remove the primer and fill in the gap. And you have replication happening on both strands. And so one strand will be what we call continuous, and the second strand will be done in sections and chunks, which lead to Okazaki fragments. So all of these details are the same. Okay, so initiation happens when you get this enzyme, right? So this enzyme has two names, topoisomerase and DNA gyrase. And I'm not sure why there's two different names. If you're interested, you can look it up and you can tell me. But the supercoiling, that super condensed three-dimensional aspect of the DNA is relaxed, and that then allows the OREC to be accessed by helicase, which comes in, binds to it, and starts to open up, and it makes what we call a little replication bubble. And at the little replication bubble, you'll see that there's a helicase at one end and a helicase at the other end, making two poles, right? And that makes two replication forks. Replication will go in both directions at the same time. Okay, so the helicase separates the double helix, leading, and you'll get two helicase that bind, and that forms two replication forks. This allows each strand of the double helix to serve as a template to be copied. What does the copying? Is DNA polymerase. But before DNA polymerase is able to attach to the template and start copying, it needs what we call a 3' end to grab onto. It needs an OH group. So we'll review in a little bit what 3' and 5' means. But for now, just kind of try to take it in that DNA polymerase needs to have a very specific type of structure in order to do its job, and the primer provides that structure for it. Okay, and then it will continue to put in base pairs in a complementary fashion until it reaches the end. But that's the continuous strand. The second strand will be discontinuous because instead of being able to keep going forward as the DNA is opened up, it's going in the opposite direction. And so it's going to run out of template and fall off. And then more template will be opened up, and then it'll start again, and then it'll run into where it last started. And this will repeat over and over again, and this leads to lots of small segments. And so then this happens over and over again on the discontinuous strand. I have an animation here which will make all of this make sense. So let's go ahead and watch that.
replication is the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having three phases: initiation, elongation, and termination. Let's begin by considering the initiation phase. For copying to take place, the double helical structure of the DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, a DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble. This establishes 2 replication forks that will move.
So this is the replication bubble. These are the replication forks. These are each going to move away from each other. And that's going to lead to your continuous strand and your discontinuous strand. Because remember, the 2 strands of DNA are anti-parallel to each other. So on one strand you have 5' end, and then on the other strand that would be the 3' end, and then it switches. Okay, so because of the orientation of the DNA molecule and because of the direction that you're opening up, and because DNA polymerase can only copy in one direction, you're going to have one of the polymerases be able to copy, this way continuously as the fork opens up, but the other one is going this way, and it's going to run out, and it's going to have to, I'll just let the video play and you can see.
Away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis. Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand. A DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome.
So those steps were initiation, and now we're entering the elongation.
The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA and encircle it, the clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring.
So the details of the sliding clamp versus the loader, we're just clumping it all together as a sliding clamp complex. So I'm simplifying this a little bit as to what you need to remember, but this still gives you a really good visual of how it works.
The ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase. The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end.
So I know you cannot see it, but you can envision that right here is that free 3' end, which is being provided by the purple primer. And so even though there is a sliding clamp here, this green polymerase is able to now come in contact with that free OH group and begin its job of putting in complementary base pairs.
During elongation, the replication bubble gets bigger as the helicases move apart, establishing 2 replication forks that move in opposite directions. The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose single strands. Let's focus on just the right-hand fork for the moment, as the processes of both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the two strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. The top strand is the lagging strand, on which just a short stretch of DNA is made by the polymerase elongating in the 5' to 3' direction. After synthesizing this short stretch of DNA, the polymerase then dissociates and a new polymerase binds at the fork to elongate the next RNA primer. So priming and elongation are happening repeatedly on the lagging strand as the replication fork moves along the DNA. Synthesis is discontinuous. The short stretches of DNA that are made on the lagging strand are called Okazaki fragments after their discoverer. During the course of replication, these fragments are stitched together. When the lagging strand polymerase runs up against a previously made Okazaki fragment while in the process of synthesizing DNA, the replicative DNA polymerase 3 is replaced by DNA polymerase 1, which is 5' to 3' exonuclease activity. This exonuclease then degrades the RNA primer in front of it, synthesizing DNA as it goes. Once the RNA primer is removed, the two sections of DNA are joined by DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment.
So when the enzyme DNA polymerase 1 comes in, which was shown in blue, go ahead and show right here, it has two functions. It has what it said endonuclease activity to remove the primer, and then it also has the polymerase action of putting in complementary base pairs. So it's a DNA polymerase that can remove the primer. So it removes the primer here, and it puts in the new DNA complementary base pairs here and fills in that gap. And then there's one bond, the bond where you start and finish, right? That will need to be sealed, and so then that's shown with the ligase. Okay. Now this next part of the video is going to show you one more level of complexity that occurs, which I'm not going to test you on, but I do want to show you just because it's pretty it's pretty cool.
DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment. While this view of the replication fork is easiest to understand, in reality the leading and lagging polymerases are coupled and move in concert with the helicase. This coupling requires one DNA strand to be looped around so that the lagging polymerase can synthesize DNA in the 5' to 3' direction while moving with the fork. We explore this coupling in more detail in a separate animation.
Yeah, so we're not going to worry about that coupling so that it moves and forms that loop and can move together in the same direction, though that is, like he said, in reality that's what happens, but it's much easier to learn the stepwise processes when thinking of it as linear. And regardless, even in that within that loop and moving in the same direction, you're still going to have the formation of the Okazaki fragments you're still going to have a lagging strand. So that loop is going to have to constantly stop, start because of this, because of the Okazaki fragments. So again, refer to the slides and see that I'm not going to test you on the coupled aspect. Okay, so for elongation, which you just saw in the video, I've written out the step-by-step pieces here for you, but I really recommend that you watch the animation several times and start putting together, having the visual understanding of it and being able to recite or memorize these steps. I also have one more animation that shows the synthesis that I want to show you.
When DNA replicates, the enzyme helicase separates the DNA strands. Single-stranded DNA binding proteins then stabilize the unwound template DNA, keeping it in an extended single-stranded state. So
now I'm not putting these single-stranded binding proteins in your slides to memorize, but I will note that they are important. If they were not present, the strands would immediately start to rejoin each other, like those hydrogen bonds between the nucleotide base pairs would start to reform. So by adding these little proteins along the single strands, it interferes with the ability to make those hydrogen bonds, and it keeps the strands separate.
So it can be copied by DNA polymerase. One new DNA strand is called the leading strand, which forms from its 5' to its 3' end by the action of DNA polymerase III. A sliding clamp forms a ring around the DNA and maintains the association of the DNA and the polymerase, allowing the uninterrupted synthesis of many thousands of nucleotides of DNA. The machinery at the replication fork includes helicase and a primase enzyme. Primase creates short RNA segments called primers on one of the template DNA strands. The RNA primers allow DNA polymerase to initiate DNA synthesis along the strand, forming short segments called Okazaki fragments that collectively form the lagging strand. A structure called the clamp-loading protein associates with a DNA polymerase molecule on both of the parental strands of DNA. The clamp-loading protein is also bound to helicase. These associations made by the clamp-loading protein coordinate the simultaneous synthesis of both DNA strands at the fork. Note that the two strands of a double helix run in opposite directions. They are anti-parallel. While the leading strand can be continuously synthesized in the direction of the replication fork movement, the lagging strand template must be folded so that its polymerase can create DNA in the 5' to 3' direction and still move in the same direction as the overall movement of the fork. Using the 3' end of a primer, DNA polymerase III lays down new DNA in the 5' to 3' direction until it reaches the previously synthesized Okazaki fragment. The DNA polymerase then dissociates from the DNA, but remains attached to the clamp-loading protein. The clamp-loading protein adds a new clamp and places the DNA polymerase at the next primer. The process repeats. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
Okay, so that's our two animations that I've given you links for to show you the steps of DNA synthesis, which I would at the top on your notes, I would write AKA DNA replication, right? Replication happens only for the purpose of cell division, right? It only happens once in each cell. So each cell will replicate its genome once and then divide, okay? It takes a huge amount of energy to do this. And as I mentioned, when doing this, the cell is not able to undergo its normal processes. The DNA will be inaccessible. So it also has to do a huge amount of work in addition to making the copy of the DNA before cell division. It also needs to make all sorts of other products and prepare for that huge process of division. Okay, so initiation, everything binds, opens up the 2 strands, you get the primer, and then the DNA polymerase 3 is able to attach to the primer and the clamp, and then it's ready to go. And of course, it's happening on 2 strands at the same time, and it will be in opposite directions because the strands are in opposite directions. And then the elongation, the DNA polymerase will continue to add nucleotides in the complementary base pair fashion. So what do we mean by complementary base pair fashion? A goes with T, C goes with G. If the template has an A, the polymerase is going to put in a T, and so on and so forth. And that is due to the binding, right? Those hydrogen pairs, hydrogen bonds between A and T and C and G, right? They don't like to bond in the other way, right? So A doesn't like to bond with C or G. And as we mentioned, the polymerase will go in the 5' to 3' direction on both strands, but because both strands are anti-parallel, this leads to the formation of one continuous strand and the other discontinuous. The discontinuous strand is also called the lagging strand, and it will lead to fragments of copied DNA called Okazaki fragments, which also means you're going to have lots of primers having to be added, and so eventually those have to be removed and replaced with DNA, because the primers, remember, are actually RNA. And so DNA polymerase 1 comes in, removes the primers, and fills it in with actual DNA, and then DNA ligase comes and seals it together. And then termination for bacteria is a little different than we see for eukaryotes, because we are dealing with a closed loop. Eventually it all will meet and have to be separated, and so we'll see that an image of that in a minute. So let's start with complementary base pairing review, right? So A will form 2 hydrogen bonds with T, and C will form 3 with G, and so on and so forth. So that's what we mean by complementary base pairing. So you have a template, and the polymerase will put in the complement as it reads the template. Remember that they are anti-parallel strands, which we saw in the video. So one end is 5', the other end is 3'. How do you know which is which? Well, when you look at the nucleotide structure, you see, starting at oxygen, the first carbon to the right of oxygen is the first carbon, then second, third, fourth, and the 5th carbon is above the ring, and the 5th carbon has the phosphate group. So when you're talking about strands of DNA, whichever end of the strand has the phosphate group hanging off is the 5' end. So see, you have a free phosphate group, nothing is attached to this phosphate. It's attached to the sugar at one end, and there's no second, right? Nothing attached to it beyond that. So it looks just like this. So this would be the free phosphate group, makes it the 5' end. On the other end, you have a free OH group. In this image, it's just shown O ... because when the next nucleotide comes in to bind, the H will have to go to make that bond. Okay, but here we see that we have a free OH group, and so then this is the 3' end, and it's so 5' to 3', and on the second strand it's the opposite direction, 5' to 3', and then we see the complementary base pairing happening here as well. And in this image, we have a nice visual that you have your your nitrogenous base A, the nitrogenous base adenosine, is a bit larger of a molecule, and it base pairs, complementary pairs with thymine, which is a smaller molecule. And we see the same kind of traits with C and G, C being smaller molecule and G being the bigger molecule. And so by having this complementary base pairing in this way, you're able to maintain a constant length between the sugar phosphate backbone. And this allows the molecule to be have the same continuous shape throughout, and it leads to the double helix. So there's a lot of properties, molecular properties of DNA that are important when looking at its structure and function. Okay, so complementary base pair, A to T, C to G. Free phosphate group is the 5' end, the free OH group is the 3' end, and the two strands run anti-parallel or in opposite directions. Okay, and this is showing the same thing, but here you can see that the 2 hydrogen bonds are labeled and the 3 hydrogen bonds here, and that the small molecule is the pyrimidine and the large molecule is the purine. So it's just giving you a little bit more maybe detail than the other image, but it is showing you all the same things that we just talked about. Okay, now here we see all the parts of replication that we were just studying. So we see the helicase opening up the 2 strands of DNA, right? We do see these single-stranded binding proteins that stabilize the protein, or that stabilize the strands so they stay separated. And we see the DNA polymerase 3 coming in and binding. What it didn't show in this image here would be the primer, which is a little unfortunate. But down here we do see the primer. So it shows the circle here is the primase, which makes the little RNA primer. And then we have the DNA polymerase attaching there, and it's moving the DNA polymerase 3 on this strand is moving to the right, and it's going to lead to a bunch of small fragments. And then the DNA polymerase on the top is moving to the left, and it'll be continuous. And so that's showing one of the replication forks. And here we can assume that this is part of a circle that's not shown, but at the other, and you would have the other replication fork, it's just not shown. And then eventually you will make a complete second circle. So you'll come all the way back around to the origin of replication, and then those 2 loops, which are each double-stranded, will need to be separated. And each of these loops is what we call semi-conservative. It has one original template and one newly synthesized strand in each of the loops, and they will get separated by the very enzyme that started the whole process, the gyrase, the topoisomerase, will separate them. And so that's what we see here. And I think, yeah, I wrote that here for you because it's a closed loop. Eventually the forks will meet, the 2 loops will need to be separated by topoisomerase. Okay, so the detail that you need to memorize is typed on these slides, and then the animations are given to you to help you understand the steps and visualize it. If there's extra information in the animation, you're not responsible for it. Okay, moving on. So we just looked at DNA replication. Now we're going to look at flow of information within the cell, which would be taking the DNA, taking one strand, which will serve as the template, and making a complementary copy into RNA. And so remember, for RNA, anytime there's an A in the template, instead of matching it with a T like we see for DNA, we match it with a U. Uracil is very similar to thymine, but we only see uracil in RNA, and we only see thymine in DNA. Okay, and so if taking this top strand as the template and making a copy of it with the RNA polymerase to make mRNA, we then can look at every 3 nucleotides when we get into a ribosome, and we call those every 3 is called a codon, and the codon codes for a particular amino acid. And so we have nucleic acid in the first 2, right? DNA and RNA are nucleic acids, and then we end up with amino acid sequence, which will form a protein. So that's why we say translation. You translate from the language of nucleic acids to the language of amino acids. Okay, and how do we do this? We do this with ribosome and with tRNAs, and so we're going to look at that process. And just like we saw with replication, we look at transcription and translation in 3 steps: initiation, elongation, and termination. So that's something you need to be careful with, because you'll see those terms initiation, elongation, and termination for all of the processes that we're looking at today. Okay, so prokaryotic transcription is fairly simple compared to eukaryotic, but they're also very similar. The difference, of course, prokaryotes don't have a nucleus, so everything's happening in the cytoplasm, whereas when you're looking at eukaryotes, we have that division of location, right? And because we have different locations, we have a few little extra details as well. Okay, so prokaryotic transcription, we see DNA transcribed to make RNA. That's the same for both. And RNA can be messenger RNA. It could be transfer RNA to bring in amino acids. It could be ribosomal RNA. The majority of the ribosome used to make proteins is composed of rRNA, but it also has other regions and components to it. We're not going to study the structure of the ribosome in this class. You can learn about that if you take molecular biology or the 4A series. Okay, so RNA, I already told you, has U instead of T, and so this is the same whether you're a prokaryote or a eukaryote. Okay. Transcription begins when RNA polymerase binds to a promoter sequence. That's also the same for prokaryotes and eukaryotes. And the promoter itself is not going to be copied into the mRNA. So that's also true. mRNA is synthesized in the 5' to 3' direction, just like we saw with the DNA synthesis. It's also 5' to 3'. So same prokaryotes and eukaryotes, that part's the same. And then transcription stops when it reaches the terminator. So all of this is true for eukaryotes. We will just have additional information for eukaryotes. Okay, the promoter is immediately before the sequence that codes for the protein product, right? The RNA polymerase can do the job of opening up the double helix itself. We don't need all the additional stuff that we saw with DNA replication. And the RNA polymerase doesn't need any special help with primer. It does its own, it does everything, right? So transcription is actually a lot simpler. Okay. And then even though it's opening up the double helix, it's only going to utilize one strand, which will be the template, right? Now in eukaryotes, we have some steps, right? So we first need to be able to access the DNA, and DNA will be wrapped around these proteins called histones. And so we have to loosen the grip so that we can get to the genes. Then we'll have the transcription just like we saw. But after transcription, we have to do some processing, which we'll look at in a little bit, what the steps of that processing are. And then it will be ready to leave the nucleus and then do translation. Okay, so the steps of eukaryotic transcription is just some extra steps. Okay, so again, prokaryotes don't have organelles. It all happens in the same place in the cytoplasm. DNA, on the other hand, is in the nucleus, and it's tightly wound. Even it's not as tightly condensed as a chromosome, but it's still somewhat condensed as chromatin. Okay, and so it still needs to be loosened before the gene can be accessed by the RNA polymerase. The RNA polymerase will make the transcript just like we saw, and then there'll be some processing to make it a mature mRNA that's ready to leave. All right. So eukaryotic transcription occurs in the nucleus. Initiation is the same with the RNA polymerase binding to the promoter. Elongation is the same with the complementary base pairing. And the termination is the same with all the pieces will dissociate. Okay, so those steps, initiation, elongation, and termination, are the same for both prokaryotes and eukaryotes. It's just some of the steps that have to happen before initiation and the steps that happen after are only for the eukaryotes, right? And so that processing that I was mentioning, we have some parts that are non-coding regions that we have to remove that actually interrupt within the gene. Okay, earlier when we talked about those short tandem repeats, that was happening outside of the gene. The gene was not interrupted. And so you simply just don't copy those short tandem repeats. They're not an issue. But with eukaryotes, we have introns that are interrupting the gene. And so we say introns are in the way. So I and I, right? Introns are in the way. They're interrupting the gene and they must be removed. And the exons need to be expressed and we want to put them together. So this is an image of what that looks like, right? So you have a gene and you have these introns that are interrupting the gene that have to be cut out. And we call that splicing. And then all the exons need to be expressed, so they're put together, and that will be the actual gene from beginning to end. But if this mRNA goes out of the nucleus, it gets chopped up, the cell will think it's foreign, enemy, okay? So we need to protect it with a 5' cap and a 3' poly A tail. The details of what those are is not important for our class, but you'll learn more about them if you take any more advanced courses. But this protects it, and now you have a mature mRNA that's ready to leave the nucleus and get to a ribosome, okay? And so the mRNA, we say, is processed. You have intron splicing to remove the introns. You have a 5' cap and a 3' poly A tail. So we have an animation that shows the steps of transcription. Shows initiation, elongation, termination, and the intron splicing, the cap, and the tail. And so this link here is talking about a eukaryotic cell. Okay, so let's go ahead and look at that.
The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed.
So remember, my little strategy for remembering which is an intron and which is an exon, or like how to remember what the function is. Introns are in the way, they must be removed. Exons are expressed, they must be put together.
And modifications such as a 5' cap and a 3' poly A tail are added. This process is called Intron Splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore.
And so remember, this has a continuous sequence that makes up the gene, and they were originally individual segments called exons, and exons are expressed. And that's all you have left in the mature RNA. It's just all the exons put together.
And enter the cytoplasm to begin translation.
Okay, and so that's the end of the first part, transcription. Let's go ahead and look at translation. So translation, you use that messenger RNA that we just made to bring in amino acids and make the polypeptide. The polypeptide is then folded and modified to make the actual protein, functional protein. Okay, how is the mRNA translated? It's in those codons. So every 3 nucleotides on the RNA sequence is a codon that codes for a particular amino acid. You have a few codons that you need to memorize. The start codon, which is always the first codon in all transcripts, AUG, start codon. Now at the end, translation ends when a nonsense codon is reached, and the nonsense codons are the stop codons, and there are 3 of them: UAA, UAG, UGA. Okay, so you need to remember nonsense codons are the same as stop codons, because there are times you'll see nonsense mutations, which means that a mutation has replaced one or more nucleotides that then led to the formation of the nonsense codon. And so that's this terminology of nonsense codons being a stop codon is very important to remember. You'll see this again when we talk about mutations. Okay, so this part is the same whether you're a eukaryote or not. It's just the location in the cell that will be different. Okay, now the specific steps of using the codon to put in the amino acids is what we're going to focus on next. Okay, so initiation has some very specific steps where first the small ribosomal subunit, so the small unit, which you see here, the small ribosomal subunit, will first then bind to the mRNA, and that will then attract the first tRNA, which will be at the start codon. So the first tRNA always has methionine, because the start codon AUG codes for methionine. Okay, so that's always going to be the first step. And then once you have this tRNA with methionine, then the large ribosomal subunit will then come and attach. Okay, so messenger RNA first, small subunit attaches to the mRNA. Then you have the transfer RNA with methionine, and then you have the large ribosomal subunit. Okay, and so then you're ready for elongation, which everything scoots over one, so the next codon can be complementary matched with the next tRNA. So we still are functioning with this principle of complementary base pairing, but now we refer to it as codon and anticodon. And again, it's every 3 nucleotides on the mRNA. And so in this way, you are bringing in specific amino acids to make a specific sequence, right? And there are 3 regions of the ribosome, the E and P and A site. And you'll see that the P site, when we're starting, we'll have the methionine, right? And then the A site is where the next transfer RNA comes in and brings the next amino acid. And then those 2 amino acids then need to be bonded together. So a peptide bond needs to be made. And it actually gets passed to the new amino acid. So you see over here, now we have both amino acids here in the A site, and then everything scoots over again. And now this empty transfer RNA goes into the E site and will be ejected. And then you're ready for your next tRNA to come in. And this repeats over and over and over again until you reach the end. So we see that the polypeptide has been, getting longer and longer. It's going to be very long, which you never see represented in animations for simplicity's sake, but the polypeptides are very long. And then eventually you get to a stop codon, a nonsense codon, which will cause a release factor to come in instead of a tRNA. So no amino acids attached, and that will cause the whole complex to then fall apart, dissociate. Okay, so stop codon is reached, a nonsense codon. The release factor will bind to the stop codon, and that will cause everything to fall apart. So here are 2 animations. One of these is the same animation that we saw for transcription, and then you can just finish watching it all the way through translation. And then the next animation is only translation. Same thing, just seeing, repetition is very important for learning these processes. Okay, and it's really important that you don't confuse DNA replication with protein synthesis. Okay, so protein synthesis is transcription and translation. You may have been wondering, like, what are these EPA sites? They're abbreviated after like the chemistry of the position. So we say like the A site, which here maybe point at these that are labeled. The A site is an abbreviation for aminoacyl site. The P site is abbreviated for peptidyl site, and then the E site is the exit site. Okay, I'm not going to test you on what does EPA stand for, but I might ask, where does the release factor bind? It binds to the A site. Where does the very first transfer RNA bind? It binds in the P site, right? So we see methionine starts at the P site. Okay, so the steps that we went over. Okay. So let's go ahead and watch the translation. We'll just watch the first video together. Oops. And actually, I want to watch the second one together. Yeah. Okay. For some reason, it's not recognizing it, so I guess we will go ahead and watch this different animation instead. Okay, sorry, these videos are different shapes than what my recording spot is, so it's going to be a little small on your view, but you have the links so you can watch them again.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So I would correct that and say the newly formed polypeptide is released. This polypeptide will then be folded, and usually you have non-protein components that are added and even multiple polypeptide chains that are put together, and all of that forms that complex three-dimensional structure that will be the functional protein. So I don't like to call it a protein until it's functional, which means it would have all those modifications and folding done. Immediately after translation, all you have is a polypeptide, which in and of itself does not have a function. Okay, so very well, that is it. So you have now gone through DNA synthesis, RNA synthesis for the transcript, and then translation for the polypeptide synthesis. We are not going to go over details of modifying that polypeptide to get to the functional protein. Again, that will be covered in more advanced classes. Okay, so a little bit to review and add on. So there are 64 codons, and we call them sense codons because they code for a particular amino acid. So we only have 20 amino acids, but 64 codons. That means that there's more than one codon that will code for the same amino acid, but you still get specific coding and specific sequences. So we call it redundant since you have more than one for a particular amino acid, but I don't want you to think that that means random. So it's degenerate or redundant, but it's still specific. And then here I put in this amino acid chart that, again, do not memorize this. This is just to demonstrate that you can make any combination of the three amino acids, and you'll see what that codes for. So GGA codes for glycine. GGG also codes for glycine, and GGC and GGU. All the GG, first and second GG, any ending to that is going to give you glycine. So it's redundant, but it is still specific. GGA is never going to give you a different amino acids. It's always going to give you glycine, and so on and so forth. Okay, so that's all this is here to show you, is that it is specific, even though some of the amino acids have, so alanine and glycine, for example, have 4 different codon combinations that will give you glycine. Others only have 2, right? So we see 2 and 4. And then methionine, there's only 1. There's only 1 start codon. And then we see the stop codon, 1 stop codon here, and then the other 2 are here. Tryptophan is also, we see here once, and that's it. So only 1 codon for tryptophan. But everything else has at least 2 that code for it. So anyways, redundant, but still specific. You do not need to memorize the 20 amino acids. You do not need to memorize any of the codons other than start and stop. And remember that stop codon is called non-sense, and any codon that codes for an amino acid is called a sense codon. And so this is a review of the eukaryotic gene expression. You should be able to tell me what's happening and what's involved, what are the different players involved, and what are the steps to go all the way through transcription and translation. So that's what I would use this image for, to test your what you remember. And then even better, start with a blank piece of paper and see if you can remember all of it. Okay, so in prokaryotes, remember there's no nucleus, so everything happens simultaneously and in the same place. So it looks a little more complicated because you have both DNA and RNA and polypeptides, right? Everything happening at the same time. I'm not going to test you on being able to like draw this structure, just the concept. No nucleus, so simultaneous transcription and translation for prokaryotes. Okay, so regulation, it was mentioned in one of those animations that for transcription to start, you need the RNA polymerase to bind to the DNA template, and that is the site of most of the control of gene expression, and we call that pre-transcriptional control, right? So before you make a transcript, can you make it or not? How quickly do you make it? How often? So pre-transcriptional control controls the transcription of a gene into RNA, right? And typically we think of mRNA when we're talking about gene expression because we're thinking about making a functional protein. But remember that genes can be made into other types of RNA for other functions like tRNA and rRNA. So this will increase or decrease the amount of transcription that occurs depending on the type of molecule or processes that happens. So for example, you don't need to make every single gene all the time. There are some genes that you only need in certain situations, and it would be wasteful to make everything all the time at high levels. We want to conserve energy and resources, right? So producing the mRNA that encodes an enzyme that's needed to adapt to a new food source. So bacteria that are using glucose, for example, the genes involved in glucose metabolism are going to be on, and they're going to be on at a high level. But once all the glucose is consumed, if there's another food source available that it has a gene for, such as lactose, then it needs to turn on those genes and start using those, and it doesn't need to keep the glucose on at a high level anymore, right? So it's going to change which genes are on, which genes are off, and how much of them are on, to what degree, right? Another example, enzymes for a different activity, maybe it's part of the cell cycle, for example, right? So we call genes that are always on, that need to be on at a fixed rate for the cell to maintain its life, would be constitutive, constitutive. Constitutive genes are expressed at a fixed rate. They're on all the time, but everything else is going to be in response to the particular environment. What does the cell need in that particular scenario to survive? Okay, so bacteria can actually cluster genes together in what we call an operon, and then the operon is controlled, either increased or decreased in the rate of transcription. Prokaryotes also have transcription factors that can bind to these operons and increase or decrease their transcription. And we'll see that transcription factors also are present in eukaryotes. We just don't have operons. And so transcription factors are molecules that bind directly to DNA to either recruit and encourage transcription or to decrease. So this binding to the DNA is going to either block the RNA polymerase, or it's going to encourage binding of the RNA polymerase to the DNA transcript. Okay, so remember, DNA is the transcript. We're trying to make RNA, so we need RNA polymerase to bind to DNA and make the RNA. Okay, so you can have what we call induction or repression. Repression is going to be a transcription factor that binds to DNA and physically blocks RNA polymerase from binding to the promoter, prevents transcription. And then you'll have induction, where you have an inducer that binds to the DNA and encourages RNA polymerase binding, helps it out. And so those are the two types of transcription factors, right? Repression or induction. In eukaryotes, we also have additional levels of control, such as the histones that we saw, right? Being able to unwrap, loosen up the DNA so it can be accessed, or keeping it tight and making it inaccessible. We also have methylation, which are methyl groups added to the DNA, which will turn off a gene. And we have ways to remove those methyl groups to turn the gene on. Okay, so lots of different types of control on whether or not a gene is transcribed. That's going to give you the most conservation of energy and resources. There is post-transcriptional control, such as destroying the transcript so that it doesn't get made into protein. And it's a little wasteful, but it's better than destroying the polypeptide, which would be the most wasteful, right? And that does also happen, and that would be post-translational, which we don't even, we're not even talking about in this course. So if you block transcription, you block translation. If transcription still occurs but you ruin the transcript, then you still prevent translation from occurring. So post-transcriptional control means you made the transcript and now you're going to get rid of it. And how are you going to do that? Well, you can make what we call a microRNA, just a very short sequence that is complementary. It'll bind and it'll signal it to be degraded. And we have, I didn't include the image, but you do have an image of this in your textbook if you want to see what that looks like. Okay, so mutations, I think I will just include another short video as this is already very long. But good job. Take a break and then come back for the last short segment on mutation and recombination. Okay.